Well, hello everyone, and welcome to the second keynote presentation of the 7th annual event in the CRISPR virtual event series brought to you by LabRoots. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's presentation. Now, on your screen are several interactive windows, and we encourage you to engage with them during the presentation. You can speak with other attendees in the live chat feature found on the bottom left. Submit questions for our speaker using the question submission window below. And you may also receive free continuing education credits by clicking the image on the bottom right and following the steps. I would now like to introduce our keynote speaker, Dr. Chase Beisel, professor at the Helmholtz Institute for RNA-based infection research. To view Chase's complete biography, click on his name below. Here to give his presentation titled Targeted DNA ADP-Ribosylation Triggers Templated Repair in Bacteria and Base Mutagenesis in Eukaryotes. Here is Dr. Chase Beisel.
Okay, thank you for that introduction, Israel, and of course the opportunity to participate in this series, and for all of those joining online either live or watching this apparently over the next year. So today what I thought I would do is tell you about some ongoing work in my group that we've wrapped up very recently. This is the title that Israel communicated, but can be much more simply described as a new editing approach we call append editing. So that's what I'll be talking through for this presentation. Now, it's just a little bit of background to get us all on the same page. This is, of course, all in the context of CRISPR technologies, which I think is the focus of this online series, which, of course, has been an absolutely astounding tool, particularly for genome surgery, by using these programmable CRISPR nucleases and easily designed single guide RNAs to direct the binding and cutting of specific DNA sequences, which is a first step towards creating different types of edits. And so, of course, as a number of notable mentions over the past few years, everything from the Nobel Prize 2020 in chemistry to many, many ongoing active trials, one of which led to the first FDA-approved CRISPR drug called Casgevy just last year. And then, of course, many applications going beyond gene therapy in realms such as molecular diagnostics, agriculture, and so on. And so one important thing worth recognizing about CRISPR technologies is, one, not only do they derive from immune systems that are found in bacteria and archaea, but these immune systems are actually incredibly diverse. And so within the current classification scheme, which actually goes up to 7 different types, we have all these proteins that encode different useful functions, everything from our gene scissors, which is what Cas9 is known for, to chainsaws, such as out of these type 1 systems, to lots and lots of different functionalities that all represent different ways of thwarting an infection by an invading virus, but of course become opportunities to create different types of technologies. Now, beyond all that CRISPR entails, it's also worth recognizing that CRISPR is not the only immune system that bacteria possess. Now, this idea that there's more out there is actually a pretty silly concept even only a few years ago, but through some incredible work by a number of groups and through tremendous bioinformatic analyses, we now know that bacteria possess well over 100 distinct immune systems, many of which we're still characterizing. And of course, each of those could be the next CRISPR, or even going back even further, the next restriction enzyme that could revolutionize technology. And so given all of these different immune systems, with CRISPR particularly at the forefront, my group has been working towards trying to understand what these different bacterial immune systems do, and of course, how we can use those for our own purposes. Now, the way we've gone about this is through a pretty established pipeline within the group. For one, we've been working to develop different characterization tools to make it easier to characterize not only CRISPR, but these other defenses, with cell-free transcription translation systems being the best example of that. But then, of course, applying those to reveal new aspects of biology related to phage bacterial interactions. And of course, developing different technologies out of those insights and discoveries, in some cases even going as far to create spin-off companies such as Locus Biosciences and more recently Leopard Biosciences. Now, given that backdrop, what I wanted to talk about today was some really recent efforts with this backdrop of CRISPR and novel immune systems to really try and push ahead into new areas of genome editing, again, following this pipeline. And so the work I'll tell you about has really been driven by 4 different people within the group. Now, this was all kicked off by an incredible PhD student who has since graduated, Scott Collins, back during my time at North Carolina State University, but is picked up and really pushed ahead by 3 different people in my current group, including 2 PhD students, Harris Bassett and Darshana Gupta, as well as a postdoc, Kostas Patinios, who's actually about to start his own group in Lithuania. And so the backdrop to this project was looking at existing editing technologies and seeing how to further expand those. And the area we've been focusing on has been in the realm of base editing. So possibly a form of editing that people even listening are familiar with, but the general idea that you could go in and create chemical modifications to DNA bases as a way of changing the identity of that base. And within those chemical modifications, that's been done in 2 general ways. One is through deamination as a way of making a slight chemical tweak to that base to trick the cell into thinking it's actually a different base, such as going from a C to a T. And then a more extreme one is glycosylation, where the base is fully stripped out, creating an abasic site, and the cell is left to figure out how to fill this in. And this too can create different bases in that gap, or that hole that's left over. And so as we've been looking at this, one thing we've appreciated is that while incredibly powerful, these represent only a small fraction of the much broader chemical space that could be tapped when it comes to chemically modifying bases. And actually, one broad area that remains to be explored is the impact of appending different chemical moieties to DNA. Now, what's nice is that in biology, there's actually a lot of examples of this, especially as part of host pathogen or bacterial phage interactions. And these can include very small modifications, as shown here, up to much larger one, as in the ADP-Ribosylation, which I'll be talking about today. And this has raised the question of, if these are introduced onto a base in a targeted way like base editing, what impact does that have? And so really that was the driving question of this project that actually had some really interesting and surprising results that we can now imagine building out into an entirely new form of genome editing. And so where we really started with this is a different defense system that was discovered only recently in bacteria, or at least was revealed as a defense system. And so this is a toxin-antitoxin system called DAR-TG, where DAR-T is a toxin that's responsible for ADP ribosylating, in this case thymines, within single-stranded DNA. And then there is an antitoxin called DAR-G that's responsible for binding and inhibiting the toxin DAR-T. But then it also has the ability to chemically reverse that added ADP ribosyl group. And it's been shown through a number of really nice recent studies that this ADP ribosylation activity can nicely thwart a phage infection, although the mechanism that activates DAR-T as part of a phage infection still remains unknown. But what was nicely through incredible work through Christoph Tang's group in collaboration with Yvonne Ahel's group, both at University of Oxford, was exactly how this ADP ribosylation gets in the way of the cell. And so what was shown is that this ADP ribosylation ends up serving as a block to DNA replication. And in turn, the way the cells can at least attempt to overcome this is by restarting replication downstream and then continuing on, but then that remaining gap left because of that ADP ribosyl group is fixed through through homologous recombination with the sister chromatid that underwent faithful repair. Now, if you do this ADP-Ribosylation widely enough in the cell or to a replicating phage, it can cause things to shut down really, really well. But we are curious as what would happen if you could just direct the same chemical modification, again, this large, bulky ADP-ribosyl group, to a specific sequence, and could that unlock different forms of genome editing? And so the way we went about this very much parallels what's been done with base editors, where we created a translational fusion between what'll be a nicking Cas9 and then one of these DART toxins, in this case, a DART2 toxin comes from an E. coli pathogen. And what was already established through some of this previous work is that this DART really likes looking for a specific motif in single-stranded DNA. It'd be described as TYT, where Y is a C or a T. And if it finds that, that's when it appends the ADP-ribosyl group to the third T within that set or that motif. And the idea is that by using or having it fused to Cas9, we can direct Cas9 to that, or we can direct DART, I should say, to that specific sequence, and that would allow DART, now recruited, to ADP-ribosylate the non-target strand that's displaced as part of R loop formation. And since the sister chromatid would normally be the source of homologous recombination, we can block that by introducing a nick in an opposite strand. Again, very much paralleling what's typically done with base editing. Now, since this, and then in doing so, the idea is that we would install this ADP-ribosyl group, and the cell would be prompted, at least we would hypothesize, to undergo homologous recombination. It would normally go to the sister chromatid, but because we've disabled that through this DNA nicking, it would look for something else in the cell. In our case, a repair template we've provided with homology arms, and that would allow for that the modified sequence in between to be faithfully incorporated through homologous recombination. And so even though this does parallel how base editors are normally used, we really saw this as an entirely distinct approach, particularly based on the editing outcomes I'll describe, warranting a new name. And so what we've been calling this is append editing, in order to capture the fact that we're appending a large chemical group to DNA, and the ADP-ribosyl group that we're using represents one of many, many different chemical moieties that could be applied with this sort of approach. Now, to further define it, given all the different possibilities, is we would call this appending an ADP-ribosyl moiety onto a thymine, which is its natural target. And so for short, it can be called ADPR-TAE. A bit of a mouthful, but we're hoping that this provides a bit of a simple naming scheme if other modifications and bases are onboarded over time. And so we first wanted to try this out in E. coli with the hypothesis that this would drive homologous recombination. So we devised a simple scheme in which we had recombined a broken kanamycin resistance gene into the genome of E. coli, with the idea that that could be repaired through homologous recombination around this target site. And in turn, that would confer resistance to the antibiotic kanamycin. And so as part of this, we can then look to see how many cells just come out the other end, reflecting how cytotoxic this approach is. But then, of course, the fraction that are resistant to kanamycin, representing the extent of gene editing. And so if we just introduce some basic controls, you can start with Cas9, the gold standard for genome editing in bacteria, where, as shown on the top, under targeting conditions, so that's what the T represents, you get this massive drop in the CFUs, which is expected since cutting the genome for the most part is lethal. And so you can end up removing cells that didn't undergo editing on their own, or those very few that managed to undergo homologous recombination with the break. And this resulted in turn in really high frequencies of kanamycin resistant colonies. Now, if you just switch to nicking alone, which again is part of our approach, the cells are much happier, but the editing frequency is much, much lower, so only a few percent. And so now when we introduce our our append editor, which again combines DAR-T and a nicking Cas9, you end up seeing, well, general cytotoxicity under non-targeting conditions. It's a toxin, not too surprising. But then the important thing was that under targeting conditions, we saw a massive boost in the fraction of kanamycin-resistant colonies. So we're really getting what appears to be editing, and we could confirm this through Sanger sequencing of individual colonies. And if you go in and just include this ADP-Ribosylation, so that's the next one, you end up with a little bit less editing activity. And of course, if you deactivate this DAR-T toxin, you get no editing whatsoever, but the cell viability is restored because you're no longer expressing this active toxin. And so we saw this as a really good example through this assay of demonstrating that the combination of ADP-Ribosylation of one strand and then nicking of the opposite strand could drive efficient templated editing. And in fact, we could confirm that even further when taking away the selection step, be biasing the overall editing frequency and result. And so if you instead just grow up the cells without kanamycin selection and then apply an amplicon of that target sequence from the population, we end up getting really high frequencies of editing similar to our prior results under targeting conditions, so that's in blue. But then if we don't target or we remove ADP-Ribosylation or nicking activity, then we also get, or we lose that editing frequency. And there are some ambiguous edits in there, but that I think goes hand in hand with nanopore sequencing as we're finding. Now, in addition, if you go in and start looking to see if this active, toxin is doing anything else across the genome and possibly causing other edits, say through base mutagenesis, we really didn't see any evidence of that. And that was the case here on the left as part of our original nanopore sequencing. It was just those templated edits. And even if we just look across the genome, we don't really see any level of at least T to other nucleotide changes. Whereas if we use a standard base editor with a cytidine deaminase, that's at the upper right, you see a decent frequency of these standard C to T edits as you would expect across the genome. At least in E. coli, as we had done here, we really only seem to be getting templated editing as a result. Now, of course, one of the issues is that this also comes with pretty high levels of cytotoxicity, something that we wanted to avoid, or at least be able to reduce. And so fortunately, Yvonne Ahill's group had published some really nice crystal structures we could use as a reference point in combination with AlphaFold to see how that compares to our specific DAR-T. And through that, we're able to identify a number of residues we could screen to see if that could reduce but not completely kill the activity of this nuclease. And so here's a large set of data showing exactly what happened based on the same assay, looking at the total number of viable cells, as well as the number that drove kanamycin resistance. And so within this large data set, it's worth pointing out that when we layered an original attenuating mutation, that's our G49D, with any one of 3 different mutations, we observed high levels of CFUs, but then we were also able to maintain that same level of editing based on kanamycin resistance as we had observed with our original construct. So signs that we're attenuating DAR-T, but without hurting editing activity. And what we ended up doing was combining those into a final version we internally call DLAA, capturing those 4 mutations, that was able to further attenuate our DAR-T, even under more sensitive conditions, for instance, when we get rid of homologous recombination. But when recombination is possible, we still get very high levels of editing. And so this ended up becoming the main one we ran with in bacteria. And so using this, we were able to test a large number of types of edits that can be created by driving homologous recombination. And so these included a large set of substitutions, where that region in red is the region that's substituted, so maintaining the DNA length, but changing all of the sequence in between. And we could get up to swapping in and out roughly a 90 base pair sequence while still maintaining high levels of editing. In this case, just by screening a large number of individual colonies to see the extent to which they're fully edited, or they represent a mixture of edited and unedited. And we were able to show that the same approach could create deletions of different size, in our hands up to 90 base pairs without pushing it farther, but also being able to insert DNA, where in this case, this seemed to be a bigger challenge for the cells, and going above 100 base pairs dropped off quickly. And of course, we want to figure out in the future why exactly this was happening. And what was nice, at least, is that we were able to go beyond also this kanamycin site that we edited many times over, and showed that we could also achieve editing at different sites across the E. coli genome, but could also do the same thing in Salmonella. So good evidence that this could represent a generalized approach in bacteria. Now, as a final point, we were also interested in what the underlying repair pathways were, and so ended up deleting a number of those and looking to see how that impacted cell viability as well as editing efficiency. And first key one was that if you delete REC-A, which is central to homologous recombination E. coli, we completely lost editing, and the cells were super unhappy with DART floating around the cell, as to be expected based on what was already known around DART. Now, in addition to that, if you mess with other genes involved in homologous recombination and different pathways like the REC-FOR pathway or the REC-BCD, the sign was that the REC-FOR pathway seemed to be playing at least some role based on reduction in kanamycin resistance, but we also saw some effect through REC-T as well as REC-J, suggesting that multiple recombination pathways are involved. But then if you delete other repair pathways, such as mismatch repair or base excision repair or nucleotide excision repair, we didn't really see any effect whatsoever. So which is to say, in this context, the editing and viability that we were seeing really seemed to be through homologous recombination. And so based on this, we see this other type of chemical modification introducing an entirely new form of editing within bacteria, at least compared to the previous base editors, which again were used in bacteria to create specific base changes at the chemically modified base. Whereas in our case, we were getting something entirely different. We were actually driving homologous recombination that could give rise to quite a flexible range of edits. And so apart from just differentiating itself from base editing, really saw this as opening up the ability to create flexible edits without creating double-stranded breaks. And that allowed us to maintain really high viability of the cells, which you don't get if you just come in and cut the DNA with something like Cas9. Now, at the same time, there are other technologies for introducing large DNA insertions, in particular in bacteria. So this is through CRISPR transposons, but they also come with the creation of these scars at the end of the insertion that come from the DNA that's recognized by the transposon-associated genes. So it's all to say that despite the many different editing technologies that exist in bacteria, we see this append editing approach offering new and improved capabilities that we hope the field will adopt and at least explore. But of course, the follow-on question, and maybe the one that many listening online were interested in, was, okay, that's what's happening in bacteria, but what about eukaryotic cells? What happens there? And of course, when you think about eukaryotic cells, you now have a more complex in a way cell, but also with entirely different sets of repair genes, or repair proteins that can kick in order to deal with these sort of DNA lesions, and maybe something different happens there or not. To explore this, we began with yeast as a simple genetic model to explore the impact of append editing in a eukaryotic cell. with the caveat, of course, that it's really good at homologous recombination and really bad at non-homologous end joining. Now, with that said, we started with the setup of trying to create a templated edit similar to what we had done in bacteria. In this case, trying to introduce two premature stop codons within the FCY1 gene on one of the chromosomes in yeast. And so following a similar setup of what we did in bacteria, only in this case we're screening individual colonies by Sanger sequencing, we did end up observing some level of templated edits using this approach. So in this case, when we're actively targeting this particular site with our fusion between DAR-T and Cas9, or nicking Cas9, we got around 17% of the colonies with the expected edit. Not great, but also it's definitely happening. And of course, you don't see this at all the moment you apply a chemically dead DAR-T. But what was interesting was what else we ended up observing as we looked at those colonies in the Sanger sequencing results. And what became apparent was that actually more often than we were getting homologous recombination, we were getting editing of that specific base that had undergone ADP-Ribosylation. And so this is that underlying sequence that represents our TYT motif that DAR-T would normally look for, and it's that third T that normally undergoes ADP-Ribosylation. And so what we're showing here is that in roughly 60% of those colonies, we went from a T to either an A or a C. So something very different than what we were observing in bacteria. And this was happening even more frequently in yeast than homologous recombination. Now, of course, we wanted to go beyond yeast into more complicated systems to see if this trend continued. And so this is what we did in partnership with Jose Alonso and Emma Nova, who are both plant researchers at NC State University, who dove into this head first. It was really awesome through that interaction where they applied a leaf infiltration assay using agrobacterium, which is a much faster turnaround time than actually trying to generate whole plants. And what they did as part of this infiltration assay is they could isolate the part of the leaf in which the agroinfiltration had taken place, and then perform next generation sequencing, albeit with the ability to ensure that each cell in that population had received our construct. So despite the fact that there was no means to select for the plasmid and the transfection efficiencies may not be the greatest, we were still able to observe base mutagenesis here as well, and specifically at the base we would expect to undergo ADP-Ribosylation. Now, in this case, it was predominantly A at that position, but then we also saw not only C, but a little bit of G that T was being converted into. And we observed this as well at an additional site, one that actually had 3 different DAR-T motifs, because figure better chance we'll see something at that particular location. And in this case, we saw a similar pattern of edits where one of those sites just seemed to be much more strongly preferred than the others. So we found this really intriguing that now in eukaryotes, we're getting an entirely different type of editing outcome compared to what we saw in bacteria. But of course, we wanted to move this into human cells as another obviously really important area for any editing technology. But again, thanks to the incredible work from Yvonne Ahel's group, we already knew up front that we were going to run into one issue, which is this protein called TARG1 that's been known for a long time, but role was really not understood until Yvonne's group showed just a few years ago that it acts very similar to the DARG antitoxin. So it has the ability to go in and strip off that exact ADP-ribosyl group that our DAR-T would introduce. So obviously a barrier to being able to introduce ADP-Ribosylation and generate this type of editing, but with the understanding that there have been other repair pathways or other proteins that have got in the way of genome editing technologies, and there have been workarounds that have been developed for those others. But at least for our purposes, we figured, okay, we should at least go in, delete TARG1, and then see what sort of edits we could create, again, using our append editor through ADP-Ribosylation. And so in this case, what we did was went into HEC293 T cells, as is commonly done for genome editing, and then specifically target a site within the commonly edited EMX1 gene, and doing this under transient transfection conditions using a single plasmid encoding our editor as well as a single guide RNA. And in this case, we're using amplicon sequencing with NGS to look at the extent of any of the edits that might be created. And so starting with similar controls as used earlier, if you were just to use a standard Cas9, of course, you get very high rates of indel formation. But then if you use a nicking Cas9, you see very, very little indel formation. And of course, none of them are giving rise to the standard base mutations that we had observed in yeast and plants. And this was the case whether we were using our standard 293T cell line or the same cell line in which we had gone in and then deleted this TARG1 gene. So now when we come in with our append editor, on one hand, we're now seeing decent levels of base mutation. So in this particular experiment, around 10% editing. But then our indel frequency was much, much, much lower than what we saw with Cas9, only a few percentage points, which was also admittedly a good sign that we weren't driving high frequencies of indels as you typically get with any technology that cuts DNA. But then, of course, these edits were dependent on having an active DAR-T as well as a nicking Cas9, in this case, at least, as we used the fully attenuated DAR-T or one in which we completely deactivated it. And then if you look at what those specific base mutations are, similar very much to what we observed in yeast and in plants, where we're getting mutagenesis of the ADP-ribosylated T based on the DAR-T motif, and that you see a general conversion in this case from a T to either an A or a C, and maybe a little bit of a G as well, and then some T's being preferentially edited over others. Now, as an aside, we still had the same worry about the DAR-T running around the cell and ADP-ribosylating DNA, which could create unintended edits. And again, something we didn't observe in bacteria because they really didn't seem to be driving this form of mutagenesis, but could be very possible in human cells. And so what we ended up doing was looking at the impact of dead Cas9, but still has the fully active DAR-T. So one DAR-T is dragged to that specific site, what you consider to be the best or worst case scenario to look at target independent editing, because DAR-T is just dragged right there, but now without that immediate nicking activity. And so we were happy to see here that if you got rid of nicking, indels, or not indels, but that base mutagenesis almost entirely went away. So it was about a 20-fold reduction in mutagenesis, which is to say it can still happen, but at a much, much, much lower frequency. And in this case, in an example where we're actively targeting DAR-T to that location. So beyond that, we ended up going beyond this one EMX site and trying a number of different sites in 5 different genes scattered across the chromosome. And so it was nice to see that we could generate these sort of base mutations at the expected thymine across just about all of these sites. Of course, knowing full well that some sites will be edited more efficiently than others, as is seen across all CRISPR technologies. But with this larger set, we could also look for certain trends that can help guide what to try targeting in the future and the types of editing outcomes we can expect. So for instance, if you now lump these together based on the specific location of that edited T, it became clear that there was kind of a preferred editing window between positions 3 and 9, if you're counting from the very beginning of your sequence on the PAM distal end. But of course, there were ones in there where editing was very good and other places where editing was very bad. So this isn't the only determinant of what makes for a good editing site. On top of that, if we looked at the specific motif that was recognized, we actually saw some interesting trends there. So namely, if it was a TCT motif, we saw generally an equal mix of the T being converted into an A as well as a C. But in the examples where it was 3 T's in a row, we consistently observed much, much higher levels of the T being converted into an A, and creates a scenario where you could actually create more of a prescribed edit to go between a T and an A, rather than it merely mutagizing that site into a few different bases. And so we see that as creating some opportunities in itself, particularly in pathogenic SNVs, including those listed in ClinVar, where you specifically want to go between a T and an A. And so now as we look at what this means in eukaryotic cells, it paints kind of another picture for what could happen by introducing a bulky chemical group like an ADP-Ribosyl group compared to the more traditional base editing approaches. And so what we showed here was that by applying this in eukaryotic cells, we were going from E to a different base, which was either an A or a mixture of A or a C, depending on the site and the organism. Now, on one hand, this is much, much closer to what's already done with base editors, particularly those that rely on glycosylation, including very recently reported thymine glycosylase that was used quite nicely to go from a T to a G or a C. And so it's interesting to see that while ours also had the conversion of a T to a C, the conversion to an A as well was something that was actually new, even though we were chemically modifying the exact same base. And so it's to say that these sort of approaches where you're introducing other chemical groups can open up new types of edits, despite what's already been nicely accomplished with DNA deamination and glycosylation. And so to round out on how we're thinking about this technology going forward, we do see it providing a number of really nice capabilities despite all that already exists in the genome editing space. And so in bacteria, really it comes with this ability to create flexible edits, but without imposing a strong cytotoxic effect on the bacteria. And again, this is when we're using these heavily attenuated versions of the toxin. And so if you can create flexible edits through non-homologous end joining, through homologous recombination, but not sacrifice the number of cells that you get out the other end, which is exactly what happens when you apply Cas9, there's some important things you can do with that. So obviously, rather than creating large edits, it allows you to access multiplexing much more readily. You could also imagine trying to create not a single edit, but a whole library of edits at one site that allows you to screen those cells. And also because the cells are preserved and not killed off most of the time, as you have with Cas9, you can do things like in situ editing with these flexible edits, where the cell that undergoes editing is going to be preserved and maintained itself in that environment. Now, in eukaryotes, of course, it's a very different outcome, where we're now going to either a T to an A, or the other extreme is just mutagenizing the T altogether. And so if we can create a T to an A edit, that does allow you to think about accessing these pathogenic SNPs that involve that exact conversion. But of course, if you can mutagenize the base in other contexts, that could allow you to mutagenize proteins in specific locations, and then be able to screen those for improved or novel functions, but already in their native context within the cell that contains them. Now, of course, within these possibilities, there's still a lot more work to be done, and a lot that we're already starting to do in-house. And so this, of course, includes figuring out how to circumvent TARG1, which is, of course, the major barrier right now to implementing these in human cells. But we also want to make this a more flexible technology. So get away from this TYT motif to something more flexible, but also get away from T as well and be able to move over to the other 3 bases. And then, of course, try out other different chemical modifications beyond ADP-Ribosylation, which again should be in the realm of possibility given all the different modifications that are already known in biology, particularly within phage bacterial interactions. Okay, so with that, I'll wrap up here and just remind you of a few takeaway points if you got nothing else out of the talk. So the first is we really see this concept of appending chemical moieties to DNA as an unexplored approach within this already well-established area of genome editing. And so in applying this, we were able to achieve templated editing in bacteria, but without taking a hit in viability to the cells that we think opens up its own set of applications. But then a very, very different outcome in eukaryotic cells where we seem to be predominantly driving base mutagenesis. Now, of course, as I pointed out in that prior slide, this is really just a first demonstration, a proof of principle, and we see a lot of different ways that this can be enhanced, expanded, and then, of course, inevitably applied to different application areas of genome editing. And then maybe as a final point, for those who aren't thinking so much about genome editing as much as pathways of DNA repair, something that's really, I'd say, been difficult in the field is being able to create these different types of base lesions in a targeted but also simple way. And so with this append editing approach, we feel like this has introduced the first way to do this, as we're already doing with ADP-Ribosylation, but by onboarding other types of chemical modifications, could explore a much wider space of chemical adducts that can be studied in a much more precise way using this approach. Okay, so with that, that concludes everything I wanted to describe. I mentioned various contributors, both within my group as well as through collaborations along the way. And beyond the funding sources, thank you again all for joining online, and happy to take any questions. Thank you.
All right, Chase, thank you so much for that informative presentation. As Chase said, we're going to take some questions now, so let's go ahead and dive into the Q&A. As a reminder to our audience, you're more than welcome to continue asking questions during this time. We may not get to all of your questions, but we'd love to get to the ones that we already have in. So let's go ahead and start with this question from our audience, Chase. Is it easy to work with CRISPR without any experience if applying for a postdoc position that requires CRISPR?
Yeah, so yeah, great question. Obviously relevant those who recognize the power of CRISPR, want to get into it, but just didn't have that opportunity through training up to that point. I think on one hand, it does depend on the type of CRISPR approach you're using. But in general, CRISPR has been fairly easy to pick and apply. And I think that's in part why it's suddenly become this widely used technology when the original insights into Cas9 and other CRISPR nucleases came really only a decade or so ago. So it is pretty incredible how easy it has been to pick up. But obviously, the more opportunities you can experience it, even in more targeted ways, the better off that's going to put you, particularly if you're looking at positions that require that as a basic minimum.
Awesome, thank you for that. Our next question is a little bit of a mouthful. I'll try to get through this, but I think it's an important one. CRISPR with Cas or GCas9 cascade, 100% efficient in blocking TARV1 effect, hence rendering ADP ribosylation of DART2, hence continuous repair of base mutation considering the off-target effects of CRISPR cascade. I can put this in your
I was able to open up. Yeah, I was able to open up the questions from my side panel so I can look at it.
Perfect.
Okay, yeah, so there's, okay, so I'll do my best with this, and there's a few pieces in there, but if I understand what Kapita is asking is, I think it's getting much more into the potential of off-target effects. And so that's what I'll try to address. And Kapita, if that's not on point, feel free to put in another question into the feed. But it is something that we've been thinking about and trying to look at as much as possible, in part because this has been a long-running question within the field, particularly as you think about applying this, for instance, as a gene therapy in patients. And so when it comes to these sort of base editing type technologies where the domain that's dragged over has its own ability to affect DNA separate from your Cas9, you really have to think about that as actually two different sources of potential off-targeting. One is Cas9 goes to a location that it wasn't supposed to, but the sequence just happens to be similar to what you were trying to target. So that's a traditional off-target. But then you could also have this chemical domain running around the cell, and it just happens to run into single-stranded DNA and can create the edit, even though Cas9 was not dragging it there. And so those are generally referred to as target-independent off-target edits. And so while the on-target edits are always something to be concerned about, and there's lots of tools and technologies out there to predict those, I think the target-independent ones are the much more challenging ones, particularly because it's really hard to predict those through computational tools. And so the closest we could get was this one data set I had shown of what happens if you just make Cas9 a dead Cas9. So it binds DNA but doesn't nick it, but then our DAR-T is dragged right there to create the modification. And there, in that case, we definitely saw a massive drop in base mutagenesis, which is a sign that if DAR-T is roaming around the cell and creates this in a rare event, very unlikely to lead to an edit, but there was still something. So it's not something we can ignore and will have to be taken into account as we continue pushing the technology forward. So long-winded answer, hopefully, to your original question.
Great, thank you for that. Our next question is there any use for CRISPR in muscle research?
Okay, so admittedly not my area specifically, but loosely familiar with everything else going on in this massive field. And as I understand it, in terms of research and understanding, let's say, the genetic underpinnings of how muscle cells function, and of course, show dysfunction as part of different aspects of disease, I'm sure CRISPR is being used heavily there as even a standard tool as a way of looking at, again, these genetic underpinnings and the impacts of individual genes, as well as even particular SNPs or even targeted mutations. At the same time, I know there's a lot of work looking at ways of tackling muscle diseases, including those that were inherited, such as Duchenne muscular dystrophy. And that's been actually a really big focus within the CRISPR community of how can we apply this technology to particularly go in and then be able to address some of these disease-causing mutations in the dystrophin gene as a way of being able to help those particular patients. So it's all to say, I'm sure there's a ton going on in this area. I know about it only superficially, but probably a few Google searches away from learning more on your own.
Awesome, thank you. Our next question, do you have any open positions in your lab?
Okay, all right, well, Nora, thanks for that. Glad I didn't scare you away over my 40 minutes of talking or so. I would say at this point in time, we don't have any active positions we're advertising, but I'm always on the lookout for strong applicants everywhere from the master's level all the way up to postdoc and even visiting researchers. And so if there's something that interests you, feel free to shoot me an email and happy to get back to you.
Awesome, thank you. We have time for several more, so let's go ahead and move on. What other DNA modifications could be explored as part of append editing?
Okay, yes. So as I kind of mentioned earlier in the presentation, we really see this ADP-Ribosylation as the tip of the iceberg of all the different types of modifications that can be introduced. And so this is everything from other versions of DAR-T to other known toxins, all the way to actually phage proteins that will adorn the phage DNA with chemical modifications to block it from CRISPR and restriction enzymes and other systems. And those represent what, or at least examples that could be explored to see what impact they have in the context of append editing. So our hope is that ADP-Ribosylation represents merely one of many different types of DNA modifications that can be explored and hopefully used in the future.
Perfect. Moving right along. What about off-target editing?
Yeah, and I feel like I had effectively addressed that one with that earlier question, so I won't belabor the point there. I'll just say that with the DNA modification domain, there's an extra type of off-target editing you have to be aware of, and something we're paying close attention to, even though our hope is that it's actually pretty limited.
Understood. All right, how about this one? Do you use nanoparticles in your lab to deliver CRISPR in vivo?
Yeah, so I think the short answer is no, only because my lab primarily works in bacteria or these cell-free systems. And then we do some work in human cells, as I had shown in the presentation, but it tends to be very simple cell culture experiments, and definitely nothing advancing to the point of being, say, in animal models. But interact with a lot of people who do this sort of work, including through collaboration. And so interacted with some people that do apply nanoparticles as part of CRISPR delivery, but again, not something that my group is doing directly. I think it's a fascinating aspect of the field, and obviously incredible as we try to make this technology even more usable in clinical settings, just not something that I'm personally advancing.
Understood. All right. Why don't bacteria also display base mutagenesis?
Right. So this was kind of one of the quandaries we've had, especially after everything that we've seen in human cells and yeast and plants. Like, why aren't we getting the exact same thing in bacteria? If nothing else, all of the other base editors, whether through deamination or glycosylation, the outcome was basically the same from bacteria all the way through eukaryotes. And so we definitely see this as a bit of a quandary. I mean, the best guess right now, obviously, is that the engaged repair pathways in all of these eukaryotic cells are not active in bacteria. You know, we originally suspected things like translesion polymerases, or translesion synthesis polymerases, excuse me. But E. coli has those. So it's still a bit unclear what really makes the difference at the end of the day, but of course, something that we want to figure out.
Perfect, thank you. Got another one in from the audience here. How ADP ribosylation-based gene editing works, considering the codon bias nature of CRISPR-Cas9 cascade that often lead to off-target effects to complete the activation of genes under mutagenesis?
Yeah, admittedly, Kavita, I'm not sure how to answer that. Maybe the pieces I can pull out of there talk about maybe the codon bias nature of our approach. Let me contemplate this one for a second. Yeah, so it's a matter of whether that codon bias, referring to it within the editor itself and how it's expressed and used, or if that relates to the that specific motif that our current DAR-T is limited to. And how about this? I'll assume the latter and run with that. But there, obviously, having that motif is a restriction on one hand, because there are just certain Ts we can't access because it doesn't fall in the right motif. But it is kind of nice because if you do have that example, DAR-T will really only focus on that third T, even though there's another T 2 nucleotides upstream. And that's one challenge of having base editors that look for that one base and don't care about context is it's going to hit all of them, even if they're all right next to each other, and you only want that one. So on one hand, in certain circumstances, it could be an advantage, but at the same time, that's why I mentioned near the end of the presentation, we really want to relax those motifs in order to be able to access more target sites.
Great, thank you so much. It looks like we have just time for one final question, so I'll end with this. As I have seen in many investigations, a big problem is the delivery of the technology. An example is when we try to make some edits within the mitochondrial DNA. In this case, can the system be applied to mitochondrial DNA?
Yeah. Great question, Vanessa. And I would say just across the board, for any editing technology, particularly as you move out of cell culture, delivery is always the bottleneck and the thing that has to be overcome. And in many regards is really dictating to date which indications are being pursued in clinical trials and which ones aren't. Now, in your case, you're talking about one where this can be in cell culture, but rather than just going to the nucleus, you're now trying to get to a separate subcompartment, the mitochondria. And so from what I've seen, like obviously this is a huge challenge. You can't just get any protein there. And if it has to drag it along an RNA at the same time, like that's really hard. And so what the field has done in that regard has really focused on, well, one, not relying on an RNA guide. So instead switching to tails or zinc fingers and then directing those in. But the idea is that those are linked or fused to one of these editing domains. So it's been done with base editors, for instance. And so there's very much the possibility that you could do this with DAR-T as a way to drive mutations there. I think the question is, what's repair going to look like in the context of a mitochondria or a mitochondrial genome versus what happens in the nucleus of the eukaryote? You could probably make some guesses or predictions based on what we saw in bacteria. But honestly, it strikes me actually as a really exciting experiment to try, just being open that editing could be almost anything. So great question.
Great. We did get one more in. I just want to squeeze this in real quick here.
Sure, happy to do that.
Great. Can DNA repair processes be activated after using CRISPR and stop the intended editing?
Yeah, so I think this is kind of the, you could call it the elephant in the room when it comes to CRISPR, but also really call it the unsung hero of CRISPR, that these DNA repair pathways are really the ones that are coming in and determining what that final DNA sequence is. And in some cases, they're doing a great job working for us. And then in some cases, they're completely working against us. And so that's what we think is happening with TARG1, for instance. Now, TARG1, I think the expectation, this is actually just a protein that's constitutively expressed, it's running around the cell. But it would make sense, too, that if there's other repair pathways that tend to be activated. And so I think it's to say that repair has to go hand in any discussion with any editing technology. And again, could be helping you or hurting you at the end of the day. And what those pathways are and what they end up doing for you is going to vary from one type of technology to the next.
Awesome. Well, Chase, thank you so much for your time and your important research on this topic. Now, before we go, I'd like to thank our audience for joining us today. And questions that we may not have time for, or didn't have time for today, plus those submitted during the on-demand period, will be addressed via the contact information provided at the time of registration. This webcast can be viewed on demand for 1 year until October 23rd, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and friends who may have missed today's live event. Feel free to continue exploring this incredible virtual space, including the poster hall, LabRoots merch store, social space, and exhibit hall. Thanks again to our audience for your interest and questions, and thank you, Chase. Until next time, goodbye, everybody.
Thank you.